Hello everyone, this is Mr. Schultz and this is AP Biology video 2.3. This video covers passive transport in the cell. It's going to cover the sections in Biology in Focus 2.5.2 to 5.3. So let's talk a little bit about passive transport, which is how cells get things in and out of their cells without energy. So that's the important thing. So first let's talk a little bit about the bilayer. And so the bilayer, trying to get my pointer to work here. There we go. The bilayer is selectively permeable. The ability of the cell to regulate what goes into and out of itself is an essential function of the actual membrane itself. So it's really important that the cell is able to control what goes in and out. Certain things it doesn't have a lot of control over, like oxygen and carbon dioxide. But a lot of other things are really important and can actually be controlled. And it's really important to control it. It's part of homeostasis, maintaining balance in the cell. So the fluid mosaic model kind of helps regulate all of this stuff. Nonpolar molecules easily pass through the membrane. So things like hydrocarbons, CO2, and oxygen are able to pass through the membrane no problem. So things like steroids can pass through, nitrogen gas can pass through. Some small uncharged polar molecules also can pass through, but sometimes they need help. So water, glycerol, urea, and ethanol can all pass through the cell membrane, but sometimes they struggle. Water struggles a little bit, which is why we have aquaporins to help them move through. Hydrophobic tail also prevents ions and polar molecules from passing through. So things like glucose and sugar can pass through, but it takes forever. So we have proteins that help move them into the cell. Glucose needs to constantly be refreshed inside of our cells because our mitochondria are constantly using it to make energy. If it's too big, it can't pass through, and ions can't pass through because they get the heads like polar objects and ions, but they can't pass through the tails. So like a sodium ion or a potassium ion or a hydrogen ion, they basically hit the tails and they bounce off and they can't get through. So when we want to move things through our cell membranes, we're going to use what's called transport proteins. The hydrophilic molecules can enter the cell through transport proteins, and there's two different kinds. There's channel proteins that act as a hydrophilic tunnel through the membrane. Aquaporin is an example of this. So here's a picture of aquaporin, likely in the video that we watched about the fluid mosaic model. And what it does is it allows water to pass through. It's like a tunnel. Okay, it's not very selective in terms of it doesn't prevent things from going back out. So water can go both in and out through an aquaporin molecule. The other one is called a carrier protein, and they hold on to their passengers and change shape in a way that moves the molecules across the membrane. And depending on what kind of transport you're doing, it could require energy or not. Today we're looking at passive energy, so this video will only cover non-energy required transport. And selective permeability depends on the barrier of the lipid bilayer and the specific proteins built into the membrane. A membrane might have more or less aquaporin or more or less transport proteins depending on the role of the cell itself. So you need to be aware that sometimes a cell will have a lot of something being passed through and sometimes it'll have very little. So an example would be our nerve cells allow for sodium to pass through them much easier because they'll have more proteins to carry them in and out or to allow them to just pass through because of the role of the cell itself. Passive transport can be broken down into a couple of different categories. Passive transport is basically the movement of things in and out of the cell without energy. One example of this is diffusion. Diffusion is the movement of particles of any substance so that they tend to spread out into the space available. In my freshman biology class, I oftentimes use the example of an aerosol scent spray. So like the ones you keep in your bathroom. And what I'll do is I'll take a bunch of it and I'll spray it in the air in one corner of the room, and then we'll slowly but surely raise our hands as the smell gets to us. Particles will naturally want to spread themselves out. Diffusion does not require any energy. The molecules move randomly, but groups have a directional flow, which means that particles will want to automatically spread themselves to be as far away from each other as possible. This is basic diffusion. And it always goes from most concentrated to least concentrated. So if you have a whole bunch of something, and I like to think of like a whole bunch of smelly freshmen, and they're all clumped together, but they smell each other and they can't handle it. So they're going to spread themselves out as evenly as possible so they don't have to smell each other as much. I know it's a silly example, but it works pretty well. A concentration gradient is a vocabulary word you're going to run into a lot during cell transport. A concentration gradient is the region along which the density of a substance increases or decreases, and no work is needed to go from a high concentration to a low concentration. Think of a concentration gradient almost like going up or downstream. If you have particles that are going from high concentration to low, they're going to spread themselves out without any energy. It's like going downstream. You can just sit in the boat and let it go. But if you want to go from an area of low concentration to high, that requires energy. It's like paddling against the current. Passive transport is any movement of particles that does not require energy to make it happen. So this whole video is just about passive transport. So no energy is needed for any of these things. So just be aware of that. All of these do not require energy. The first major type of passive transport other than diffusion, and diffusion would be things like oxygen and carbon dioxide, but this is a little bit different. Osmosis. And everybody's like, hey, the movie Osmosis Jones. And I'm like, yeah, but what is osmosis? So, osmosis is water diffusing across the membrane from an air region of low solute concentration to higher solute concentration. So, osmosis is the diffusion of free water across a selectively permeable membrane. Water always moves to spread out solutes. Everything else goes from high concentration to low concentration, where it spreads itself out. Water likes to spread everybody else out, so it will always go from an area of low concentration to high. And when we look at this, we're going to talk about tonicity. It's the ability of the surrounding solution to cause a cell to gain or lose water. And it depends on the solution concentration and the membrane's permeability. So if we look at and put in a cell membrane or a selectively permeable membrane, and we have on one side lots of little solute particulates, and on this side even more solute particulates, what we would expect is if the little particles could move past this membrane, we would see particles move this way into the empty side. However, if only water can move past this membrane, water is going to move to spread the particles out. And so instead, what water does is it goes to the other side, and it spreads all these guys out. So water does the opposite of all the other particles. Its diffusion is backwards. So osmosis is kind of a reverse diffusion. Kind of, not quite. When we talk about water potential problems in class, we'll actually do them together in class. But if you have more questions or you want me to make a video reviewing them, please let me know in class and I'll actually add some water potential problems as well as a video. So, a little bit more about osmosis because there's a lot to go with this. There are three types of solutions when we talk about osmosis: isotonic, hypertonic, and hypotonic. In an isotonic solution, it is the same on the outside of the cell as the inside of the cell. This means that the solutes on the out and in are the same. In a hypertonic solution, there is way more solutes on the outside of the cell than on the inside of the cell. I kind of like to think of hypertonic as like if you work at a daycare and you have lots of kids running around you, they're all hyper, and so they're all moving around constantly. In a hypotonic solution, what you have instead is more solutes inside of the cell than outside. And I think of it like a hippo. Hungry hippos, they have all the solutes inside of them. So hypotonic, there's very little solute outside and most of the solute inside. Hyper is the opposite. More solute outside than inside. And then iso, same on both sides. Now, cells have to do what's called osmoregulation. It's the control of solute concentrations and water balance. And some organelles actually have a specialized, some organisms have a specialized organelle that helps them do this. Paramecium have this tiny little vacuole inside of it. It looks kind of like a star when we look at diagrams of them. And it's a contractile vacuole. And it allows it to like force water out of its body. It lives in a freshwater environment, which means that water is constantly trying to get into the cell to spread its particles out, its solutes. And so what the cell does, the paramecium, is it constantly is pumping water out of its cell. So that's a really cool example of osmoregulation. Our cells do it too. It's one of the reasons why we gargle salt water, so that we can reduce the pressure on the cells in our throat when we have a bad cold. So water balance with the cell wall. So this is plants, okay? Cell walls create what's called turgor pressure that hold the cell together. Plant cells need a lot of water to hold their structure together, whereas we have skeletons or exoskeletons to hold our bodies together. We have a skeleton, insects have exoskeletons. Plant cells in a hypotonic solution. So if we put a plant cell in a hypotonic solution, what's going to happen is there's more solute inside the cell than out. And water, being water, wants to go inside the cell to spread it out. And we call that turgid. It's pressure that keeps a plant stable. So that's why you want to keep watering your plants so that they can stay upright. Now, if a cell is in an isotonic solution, which means it's the same outside as inside, the cell is going to have a balance, which actually for plant cells is a bad thing. And so the cell actually ends up being a little flaccid, which could cause the plant to droop. Plant cells that are put in a hypotonic solution, like if you put some plants in salt water and they're not used to it, they're going to undergo what's called plasmolysis. And what happens is the plant itself will wilt. The actual cell membrane will pull off of the cell wall and the whole kind of structure kind of collapses in on itself. And this is when we see like plants die. So if you were to water like crops with salt water, what's going to happen is it's going to pull the water out of the cell and all the plant cells are going to droop and kind of suck in. Now, if you don't have a plant cell wall, you're going to do something a little bit different. And so we're going to see those words in a little bit. Sometimes passive transport needs help by proteins. An example, anytime we have passive transport that is helped with proteins, we call it facilitated diffusion. It's still diffusion, it's still going with the gradient, but it needs help. Facilitated means help. So facilitated diffusion is a transport of polar molecules and ions with the help of a protein. No ATP is needed for this, so this is again just passive transport. And there are two types of proteins that can help with facilitated diffusion: ion channels and gated channels, which we've seen already in this video. So just be aware of that. Oh, I made a mistake. I apologize. There are two types of channel proteins: ion channels, which allow for salts to pass through, like or hydrogen ions, protons, and gated channels, which are kind of controlled and generally require some signal protein or signal molecule to open them or close them. And then there are carrier proteins. So there are two big kinds. Channel proteins are broken up into two groups. I'm sorry about that. You should note, facilitated diffusion speeds transport of solute by providing an efficient passage. It does not alter the direction of transport. It cannot cause things to go against the gradient. In order to do that, you need to have active transport. So what I realized is that this video, I mentioned that there's with a cell wall and without. So this is without a cell wall. If you put a cell in a hypotonic solution, if you were to take your cells and put them in pure water, what would happen is the water around the cell would rush into it to spread the particles out, and the cell would basically explode. It more or less just kind of pops and just kind of oozes. But we call that lysed. If you put a cell in an isotonic solution, which is what our IVs are at the hospital, this cell will be happy. It'll be normal. It's the same on the outside as it is on the inside. And if you put cells in a hypertonic solution, like a saltwater solution, it pulls the water out of them. And so what we see is the cells will shrink, and then they won't function correctly. So when we talk about IVs, they're always an isotonic solution. Hypotonic, your cells would explode. Not good. Hypertonic, it would suck all the water out of your cells and you would get dehydrated. Also not good. So please be aware that when we talk about cells being put into these solutions, in plant cells and animal cells, different solutions are more important and healthier for each type of cell. So this is the end of the passive transport video. If you have any questions, please feel free to ask me in class. I'm more than happy to explain things, draw diagrams, or do anything else that could help you understand this material for the test.